Okay, here we go. The Chapter 12 study guide, broken up into the 4 sections. So let's go ahead and review this. Try to do this as efficiently as possible. I think we have a pretty good system going here. Please let me know if there is a better way, because I'm always open to suggestions as we move forward. Okay, let's see here. I think that would be just the easiest way to do this. So here we go. Number one, part of the nucleotide structure. Here we have a phosphate group. Number 2 here, we have a sugar. Now this can be, depending on if we're looking at DNA or RNA, this can be a deoxyribose sugar for DNA or a ribose sugar for RNA. All right, and then here we have the base. This would be the A, the T, the C, the G. And if we're dealing with RNA, we would be, there could be a U here instead of T. Sorry about that. Number 3, these types of bases are purines. Let's see here. Number 4, this represents a guanine base. Number 5, these are pyrimidine bases. And number 6, here we go, represents cytosine. So notice the A and the T, they bond together. One's a purine, the other's a pyrimidine. Not going to really worry too much about the difference here. Just notice the structural difference, how there's essentially 2 rings, if you will, for the purines and only one, a singular ring here for the pyrimidines. All right? Okay, let's go ahead and look. So here's our word bank. We'll go one by one here. All right. Okay, number 7, adenine, A, guanine, cytosine, and thymine are the 4 nitrogenous bases in DNA. Number 8, in DNA, cytosine always forms hydrogen bonds with guanine, C and G, G and C. Number 9, the sequence of nucleotides carries the genetic information of an organism. Number 10, Chargaff's data states that the number of purine bases equals the number of pyrimidine bases. Number 11, the twisted ladder shape of DNA is called a double helix. Number 12, DNA is the genetic material of all organisms. Number 13, the pyrimidine bases have a single ring structure, as I was saying before. Number 14, the purine bases have a double ring structure. Number 15, DNA and RNA are the 2 nucleic acids found in living things. And number 16, DNA supercoils to make up the structure known as a chromosome. Moving on, section 2, some matching. See if we can do this here. Number 1, unwinds in multiple areas as DNA is replicated. E, eukaryotic DNA. Number 2, parental strands separate and serve as templates for new strands of DNA. That is A, semi-conservative replication. Number 3, the DNA of prokaryotes. F, circular DNA. Number 4, keep the strands of DNA separate during replication. So what does that? C, single-stranded binding proteins. Number 5, elongates as DNA unwinds and is replicated continuously. D, leading strand. And number 6, if you can't figure it out, unwinds the double helix. All right. Obviously, that would be B, DNA helicase. Okay? All right. If I can get this. Okay, I think you can see everything now. But I can't. Hold on. What's going on here? Okay, here we go. Okay, here we go. Number 7. Okay, and 10. Yeah. Okay. Number 7, the leading strand. Number 8, this little bubble, if you will, DNA polymerase. Number 9, the parental DNA. Number 10, the Okazaki fragments. Number 11, DNA ligase. And number 12, DNA polymerase. All right, there you go. So there you have it for the figure. All right, section 3, true, false. I don't know. There we go. Let's see here. Number 1 is true. Number 2 is false. Oh, good. Here we go. Number 3, express the following sequence of DNA nucleotides as complementary mRNA codons. Okay, so here we have, this is just going from DNA to mRNA, also known as transcription. So we have AUG, GCU, AAU, UGU, UGA. All right. Oops. From that, write the specific amino acid or code that each mRNA codon from statement number 3 above represents. So we break this down into codons. So every 3, AUG, we look this up. I'll get rid of this. I'll get rid of this. Good. Okay. So AUG, AUG, start codon or methionine. The next one, GCU, GCU, alanine. Next one, what do we say, GCU? AAU, AAU, asparagine. Number, let's see, that was AAU, AAU, UGU, UGU, cysteine or cytosine. What do I have here? Cysteine. And then last one, UGA, that's a stop codon, UGA, stop codon. What are the stop codons and start codons? Start is AUG, stop is UAA, UGA, UAG. Good. So hopefully that helped you go through transcription, and then from transcription, use the mRNA sequence, break it up into 3s, use your codon chart or wheel, and then translate it to the amino acid sequence. And that's it. All right. What do we have here? Some fill in the blanks, word bank. Once the mRNA is synthesized, it leaves the nucleus and enters the cytoplasm. The 5' end of the mRNA connects to the ribosome, where it codes and where the code is read and translated to make a protein in a process known as translation. In translation, tRNA interprets the mRNA codon sequence. Once the mRNA is associated with the ribosome, a tRNA with the anticodon, CAU, will bind to the mRNA start codon, AUG. Okay. Last one, section 4. Thanks for making it this far. True, false. I'll just let you, if you want to pause it. True. Number 2 is false. It should say an operon where the italicized word or words are. Number 3 should be transcription. Number 4, so 3 is false. 4 is true. And 5 is false. It should say body plan. All right, let's see. Where's number 6? We'll get there. We'll get there. Here we go. Here we go. Okay. This figure, number 6, record the mRNA codon sequence that would result from a substitution mutation of A instead of G in the amino acid alanine in the above protein. So find alanine. Here's the protein labeled there. We'll find alanine, ALA, right there. And it says there should be an A instead of a G. So everything prior to that is the same. Everything after that would actually be the same. The only thing that's changing is this G is becoming an A. So it should say ACA, and that's right here. That changed to ACA instead of GCA. All right? That's all that is right there. And this little table here, I'll just show you that. So what type of mutation do we have here? Just some recognition. Here's the mRNA sequence. There's the mutated sequence. And what type of mutation is? The first one up is substitution, because we have CCG and here we have CGG. All right, everything else is the same. Number 8 is also a substitution, because here we have GAA and here it's GAU, but everything after, notice, is the same. Frame shift, here we have the strand. However, if you look, there is something missing, okay? So it should say UUC for the second codon. Here it's UCC because one of those U's has been deleted. So now everything shifts, hence the name frame shift. Everything shifts back that way, one spot. And that's why we're left with only a 2 base codon at the end. And that's why there's that little asterisk there. All right, so notice it changes everything after the mutation. So instead of UUC, now we have UCC. Instead of CCU, now we have CUU. Instead of UAA, now we have AA, and that's it. All right, so that's it for the Chapter 12 Study Guide review. This is Mr. Case, signing off.